Hello, Year 13. Here we are. We are ready to start talking about respiration. Okay, apologies first. There's an awful lot going on here today. So that's the sander you can hear now. In a minute, you'll probably hear the drill. You might hear the dog. Anyway, I'm going to plow on regardless. So the way I'm going to approach this is I am going to present PowerPoints as if I was stood in front of you in the classroom. But what I'm also going to do is I'm going to load up the PowerPoint so that you can see that on its own. So if you want to just read the PowerPoint without listening to me, or if you want to go back and have another look at something, you can do that. There are videos embedded in most of these PowerPoints, and the easiest way to watch those is to actually be watching the slideshow. So you need to press the present button in order to do that. Okay. So if you are watching this and not watching the video, if you're looking at the PowerPoint, make sure you press the present button and then you'll go into full screen. I apologise if I am, singing to the choir here. All right, okay. This is respiration, okay? It's not a single reaction. It is 10 stepwise reactions that lead to the breakdown of glucose and the formation of 2 pyruvate molecules. So every single molecule of glucose produces 2 pyruvate molecules. Okay, and like I say, it's 10 steps. The good news is that you don't have to learn all of this for your A-level studies. But I'm putting it up there, and I will refer to it 2 or 3 times for reasons that will become clear as we move along. Okay, so let's go to the top. Respiration, 4 main stages that are linked. So glycolysis leads into the link reaction, which leads into the Krebs cycle, which leads to the electron transport chain. And the whole point of glycolysis is basically to turn all of that all of that energy in glucose into ATP, into a little package of energy that can be used. Okay, so here we are. We are in the very first stage, which is glycolysis. Our learning outcomes today are to be able to describe the first stage of respiration, which is glycolysis. Okay, so glycolysis happens in the cytoplasm of the cell, not in the mitochondria yet. Okay, so the first stage of respiration happens in the cytoplasm or the cytosol. It's the only step of aerobic respiration that doesn't require oxygen. So glycolysis will happen in the presence or absence of oxygen. And we're going to come and have a look at aerobic respiration when we've finished. We're going to come and have a look at anaerobic respiration once we've finished aerobic respiration. But glycolysis happens in both forms of respiration. Okay, looking at the root of the cell, glycolysis does what it says on the tin. It cuts a sugar, and that's what we're doing. We are basically breaking glucose down into 2 molecules of pyruvate. And this is basically what's going on in glycolysis. Glucose is first phosphorylated twice to form a hexose phosphate. Remember, hex means 6 and ose means sugar. So this is a phosphorylated 6-carbon sugar, hexose phosphate. That hexose phosphate splits in 2 to form 2 triose phosphate molecules. Okay, and remember the prefix tri, think about your Welsh, means 3 carbons, and ose means sugar. So we've got a phosphorylated 3-carbon sugar. Now, these 2 triose phosphate molecules need to be dehydrogenated, which also is oxidized. So they're going to lose electrons, they're going to lose hydrogens in order to form the 2 molecules of pyruvate that mark the end of glycolysis. In this process, because remember, if something is oxidized, something else has to be reduced, we produce reduced NAD. If you remember, or if you've looked at my introduction PowerPoint, NAD are those electron acceptors. So they're going to be carrying the hydrogens. And the nomenclature or the wording that the WJC use is when NAD has accepted those electrons, has accepted those hydrogens, it's called NAD reduced or NAD subscript RED, which you can see I've just written here. During this process, there is enough energy released to phosphorylate 4 molecules of ADP. So we are actually producing a little bit of ATP in glycolysis, but not much. Okay, so this is basically the sort of 3 reactions that you need to know for glycolysis. And this is sort of like the description of what's going on. All the way through the first few steps of respiration, we count carbons. Okay, so you'll see this on my slide, and we're just sort of looking at what's happening to the carbon molecules, to the number of carbon atoms in a molecule. So here we've got a 6-carbon sugar glucose, which turns into 2 3-carbon molecules, which are pyruvates. Okay. So what's going on? First of all, you'll notice that we're actually putting ATP in at the beginning here. The first step of glycolysis is often called the investment step, and you'll hear people say, oh, you've got to spend money to make money. You've got to put a little bit of energy in to get glycolysis going, okay? And what that basically does is it phosphorylates glucose. Those 2 ATP molecules give up their phosphates, phosphorylating glucose. Phosphorylated glucose, or this phosphorylated molecule, because it's no longer glucose, this phosphorylated molecule is more reactive than glucose. Glucose itself is full of energy, but it's not particularly reactive. So by phosphorylating it, we make it more reactive. So that means that we reduce the activation energy required to get the whole process going. Okay. So then we have where we've got a 6-carbon sugar that has been phosphorylated twice. 1, 2. Okay, so I'm going to have a little bit of a whine about the WJC here. A number of occasions through year 13, the WJC use slightly different terms to the terms that you will find generally if you're doing wider research. And they do this with the best will in the world, because what they're trying to do is make it simpler for you. But the problem is, if you look elsewhere in the literature or you watch other videos, you will find different terms. Okay, so when that happens, I'll flag it up for you, and this is the first occasion. So the WJC refer to the molecule that is produced when glucose is phosphorylated as glucose diphosphate. Okay, elsewhere in the literature you will see it referred to as fructose 1,6-bisphosphate. Okay, and this is a picture of it, so you can see this is glucose. This is the fructose 1,6-bisphosphate. So you can see that it's a fructose structure, not a glucose structure. If you remember back to the first term of your AS level studies. And if you also remember the nomenclature, the glucose 1,6 is telling you where it's phosphorylated. So it's phosphorylated on carbon 1 and carbon 6. Okay, so glucose diphosphate is the answer that you're going to give in the exam, but if you look elsewhere in the literature, you might find it referred to as fructose 1,6-bisphosphate. Okay, so now we've got this 6-carbon sugar and it's been phosphorylated, so it's nice and reactive, and then we need to split it in 2, because remember, we need it to be smaller because it needs to get into the mitochondria. Glucose is too big to get into the mitochondria. So what happens, this is the lysis step. So this is where the 6-carbon sugar is split into 2 3-carbon sugars. So we end up with 2 triose phosphates, 2 phosphorylated 3-carbon sugars. Okay. In the final step, in order to produce pyruvate, we need to dehydrogenate. We need to remove hydrogens from the triose phosphate. Okay. And remember, when we dehydrogenate something, it's also oxidation. So these triose phosphate molecules are being dehydrogenated. They're being oxidized. And if you think back to redox reactions, if one molecule is being oxidized, another molecule has to be reduced. So if this molecule is giving up its hydrogen, the hydrogen atom has to go somewhere, and it goes to NAD. If you remember back to the first video where I talked about all the terms, NAD are electron acceptors, and they're basically accepting the electrons and the hydrogen atoms. And when they do that, they become reduced. Okay, and we call them, well, the nomenclature that the WJC use is NAD reduced, and you can see that here. Please ignore this H, okay? I'll explain more why some literature refers to NADH a little bit later, but it's just NAD reduced. Okay, so NAD is being reduced and triose phosphate is being oxidized or dephosphorylated. And these reactions produce enough energy to phosphorylate an ADP molecule, or to phosphorylate 2 ADP molecules. So in this step here, if we're just looking at this side of the reaction, 1 3-carbon sugar is dehydrogenated to produce pyruvic acid or pyruvate, and that process releases enough energy to phosphorylate 2 molecules of ADP to produce ATP. Okay? Now, really important when we're studying respiration is that this reaction happens twice for every glucose molecule, because we're producing 2 triose phosphates. Okay, so when you're counting your carbons, or you're counting your ATPs produced, or you're counting the NADs produced, from this point onwards, you need to double it up for every set of reactions. Okay, and that will become much clearer as we move along. Last little note before we move on is this pyruvic acid. Okay, so basically pyruvate and pyruvic acid are the same thing. Okay, acetate and acetic acid is the same thing. The H just refers to when something is in solution. Okay, so if you see those 2 words, just be aware that it's the same thing that we're talking about. Okay, so those of you who are chemists are going to look at this, and you are going to realise fairly quickly that you can't jump from this molecule to this molecule in one reaction. Okay, it's not going to happen. It doesn't work. And this is why I have put these 10 steps in. So for those of you who are chemists and are kind of upset by the fact that we're suddenly producing this molecule from nowhere, it's not actually being produced from nowhere. We're just skipping some of the steps to make it a little simpler, a little easier for you to remember. So the 3 steps that you need to know, if I use my mouse here, this is the first step, the phosphorylation of glucose. This is the second step, which is the splitting of the 6-carbon hexose. And then this is the third step, which is the dehydrogenation or the oxidation of the 3-carbon sugar. Okay? But each of those steps involves multiple reactions that you don't need to worry about, but if you're confused about where certain molecules are coming from, this explains it. Okay? And the summary at the bottom here, we have put in 1 molecule of glucose, we have put 2 molecules of NAD in, and we've put 2 molecules of ATP in. Remember, the ATP provides the energy at the beginning and also provides those phosphate molecules at the beginning. What we get out is 2 molecules of pyruvate, 2 molecules of reduced NAD, and 4 molecules of ATP. This is quite important. We look at the net production here. Okay, because we've put 2 in at the beginning, although we've produced 4, we've actually only got a net production of 2 because we've put 2 in at the beginning. It's kind of like net profit and gross profit. Okay. Also heat. Remember, sort of like there's always a little bit of heat produced in any reaction. That's no different with respiration. It's why we're 37 degrees. It's why we run warm, is because of the heat released during respiration. Okay.